So the second speaker for today is Mike Hammerling from Mike Jewett's lab. Mike got his PhD in Jeff Barrick's lab at the University of Texas at Austin, and now he's postdoc at Northwestern University in the Jewett lab. He studies evolvability and how that relates to the genetic code. And it's all yours, Mike.
Okay, can everyone hear me?
Yep.
You can hear me. Okay, great. Let me share my screen. So thank you, Kate, for that introduction, for having me today. Can you all see my screen?
Yes.
Okay. So I was happy that Paul mentioned that cell-free systems were so important in elucidating the genetic code. This is something we always talk about. And we kind of hope at the Jewett Lab that cell-free systems can sort of help determine the future of the genetic code as well. So what I'm going to talk about today is in vitro synthesis and directed evolution of the ribosome using ribosome display. So in the Jewett Lab, as Paul talked about a little bit, we like to engineer biological systems in vitro using cell extracts mostly. The core of this has always been cell-free protein synthesis, ribosome engineering, and producing non-standard biopolymers, particularly proteins with non-canonical amino acids. But we've also recently delved into understanding protein glycosylation and metabolic engineering using cell-free systems. I generally work on these two core issues, and I'm going to talk about that today. So we'd like to imagine how you might use cell-free systems to translate alternative polymers using the ribosome. We're all familiar with how the canonical genetic code works. The ribosome decodes messenger RNA using the standard and universal genetic code, creating proteins. Now, over the last several decades, technology has been developed which enables us to incorporate non-canonical amino acids. That means L-alpha amino acids that have non-canonical side groups, R groups. And this has allowed us to create functionalized proteins and to study the evolution of organisms with non-canonical amino acids. But we'd really like to take that a step further. And what that means to us is actually evolving the ribosome and the translation apparatus generally to accept exotic monomers. This means things with not just with different side chains, but with non-canonical backbones. So you could imagine beta or gamma amino acids. Some work has already been done evolving the ribosome to accept beta amino acids, as well as more exotic things like D amino acids, alpha keto acids, or polypeptoids. This is really challenging for the ribosome. It's been shown that you can make a few bonds, but after that, the ribosome sort of chokes on these monomers and aborts translation. It has billions of years evolving to incorporate just L-alpha amino acids, so it's a pretty big challenge to try to make it do something different. Just stepping back a little bit and talking about how I fit into this. In my graduate work at the University of Texas with Jeff Barrick, I studied evolvability of organisms that use these expanded genetic codes. So the question I was interested in then was, if you give an organism a 21st amino acid, will it adapt to use it while it evolves to higher fitness, or will it not? And what we found was that if the genetic code is efficiently incorporated and with high fidelity, that organisms can readily use non-canonical amino acids to improve their fitness. I'm interested in looking at this question from a different angle now, namely, is the genetic code itself evolvable, and what will it take to achieve that goal? So this is a project I'm going to talk about today, where I tried to develop a directed evolution platform in vitro for the ribosome. I just want to talk about translation engineering a little bit. This is really a complex problem. We know that the ribosome is at the core of translation, but in reality, what the ribosome is doing is orchestrating all of these different translation factors and tRNAs in order to actually decode messenger RNAs. So you have to really engineer translation, you have to be able to engineer amino acyl tRNA synthetases. You have to be able to engineer all the translation factors like EFTU, which is going to deliver tRNAs to the ribosome, or the release factors, which terminate translation. And you need to have a complement of tRNAs, which are orthogonal and efficient and can actually decode messenger RNAs in a unique way. So there are many problems and difficulties associated with this. And we think that for this reason, actually, this is one of the reasons why cell-free systems can really be a powerful way to tackle this problem. First of all, in vivo engineering of the ribosome faces some challenges. There are many dominant lethal genotypes. which are translationally competent, but will kill the cell if they are expressed in vivo. Some of these unusual monomers may have limited cell permeability, or if they do enter the cell, they enter cellular metabolism or are discriminated against and removed from tRNAs if they're charged to them. Finally, ribosome fitness is obviously very closely linked to cellular fitness. If you change the ribosome and it is detrimental to cellular fitness, it may not matter if the ribosome that you have created is doing the task that you want to do, because you're killing the cell in the process. So for these reasons, and it's for some positive reasons, including in vitro systems allow you to precisely manipulate the translational environment. You can add and remove translation factors at will. You avoid transformation bottlenecks in bacteria, which will reduce your library size when you're doing selections. And in vitro systems also enable rapid experimental turnover and high throughput. So we can do experiments and engineering experiments in 96 or 384 well formats and really speed up the design build test cycle. So with all that considered, I wanted to sort of tell you about our platform for building ribosomes in vitro, which is really the cornerstone of how all this works. So this is the ISAT system. The ISAT system was developed by Mike Jewett at the end of his postdoc, and some people in the beginning of his time at Northwestern University, some grad students and postdocs as well. And this is our system for building ribosomes in vitro. So the way that this works is we create a crude cellular extract. And the way this is different from a normal cell extract system is we spin out all the ribosomes using an ultracentrifugation step. We remove the ribosomes, but most of the other important stuff is still in the extract. So then into this extract, we can take a plasmid which encodes the ribosomal operon. We can express the ribosomal RNAs using T7 RNA polymerase, and then also express a reporter such as superfolder GFP. And this is all done in the presence of TP70, which is the name that we give for the total complement of ribosomal proteins, which are purified off of ribosomes. So perhaps surprisingly, when you express these ribosomal RNAs in the presence of this complement of proteins and cell extract, they will actually assemble into translationally competent ribosomes in that cell extract. And then we can sort of assess how successful our ribosome assembly has been by translating this reporter protein. So with all of this work done, I wanted to try to expand this, this ribosome building platform into a ribosome-directed evolution platform. So this is where the next acronym comes in. This is RISE. So RISE basically took the ISAT system and coupled it with ribosome display to come up with to develop a platform where we can do directed evolution on the ribosome. The way that this works is you take a library of ribosome sequence variants encoded on a plasmid. You put them into our ISAT system as I described, and you express these ribosomal variants. But the special thing about RISE is the messenger RNA that we give the ribosomes to translate encodes a selective peptide. So what we generally ended up settling on is the 3X flag tag. And this messenger RNA does not have a stop codon. So when the ribosome reaches the end of the messenger RNA, it will stall and it will form this ternary complex. Ribosomes which have successfully formed ternary complexes can then be captured by anti-flag magnetic beads. We wash away all the unsuccessful ribosomes. And then we capture and purify the ribosomes which have succeeded in our challenge. We reverse transcribe them and characterize them, and then ultimately we put them back in a plasmid, and we can iterate on this RISE cycle and in each round further enrich the most active variants. So with this conceptually at hand, we wanted a sort of a target selection that we could do that would really allow us to characterize the system in depth and get a good idea for how we can modulate the stringency of selection and efficiency and really optimize it to the best of our ability. So what we settled on was a selection that's been done in the past on the ribosome, which is resistance to the antibiotic clindamycin. Clindamycin binds to the 23S ribosomal RNA and prevents translation. So you can see here in cyan, we have clindamycin depicted. The white sticks are the library that we chose to create. So this is positions 2057 to 2062 of the 23S. In the gray sticks here, you can see these positions also form contacts with clindamycin. But the reason we went with the library we did was that, first of all, there's a history of it. This library has been tested before. And further, as a further justification, you can see if you superimpose the nascent polypeptide on top of this, you can see that these gray positions, which form contact with clindamycin, are quite close to the peptidyltransferase center. So we were worried that by changing these, we would disrupt peptide bond formation, whereas the positions that we chose kind of extend away from the peptidyltransferase center into the exit tunnel. And so we thought that these may be a little bit more amenable to modification. So we put this library into our RISE system. This is a depiction, it's called a donut plot. It's basically a pie chart with the center cut out. And each genotype is depicted as a slice. As you can see in the initial frequency, our library was biased towards wild type. That's going to be okay, as I hope you'll see in a minute. And most of the other genotypes that were present at a low enough frequency that you couldn't really depict them as a single slice. So they all fall into this sort of gray bin of things at very low frequency. You can quickly see after one round of selection that we start to get a lot of enrichment of certain genotypes. We've lost about 3,000 of our initial genotypes, and we're getting enrichment of a handful of them. As you proceed through the selection, we lose more and more genotypes, and you can see that, first of all, by round 3, wild type is greatly reduced in the population, and by round 4, it's over here. It's maybe perhaps the 40th or 30th most prevalent. So it's now being removed from the population. And the top perhaps 7 genotypes now make up a majority of the population. So after only 4 rounds, we're able to enrich for just a handful of genotypes that look like they're the most active. And if you kind of take a look at these genotypes, a lot of them look very similar to each other. So this is encouraging. We think we're selecting genotypes that perhaps have similar sequence because they have similar function. So as another way of looking at this, I took the top 10, 20, 30, and 40 genotypes, and I just plotted for each genotype its frequency across each round of selection. So you can see that for the top 10 genotypes in green here, these genotypes increase in frequency throughout the course of the selection all the way through round 4. Whereas with some of the more moderately fit genotypes like 20 through 40, they initially increase in frequency, but as the average fitness of ribosomal variants in the population increases, eventually these genotypes are no longer more fit than average. And so like for the top 20, by round 3, they're sort of starting to decrease in frequency in the population, whereas for genotypes 20 through 40, by round 2, they've most of them are below the average fitness, and they're being reduced in frequency now. This is what you would expect, basically, if you're applying a consistent selection pressure to the ribosomes. And so this is encouraging that we're not just sort of selecting for phenotypes that we don't understand. We're not selecting erratic things. We're getting a really constant selection pressure and a consistent behavior of each genotype throughout the selection. So of course we want to characterize these genotypes, or this pool of genotypes. So we assess the ability of the selected genotypes to produce superfolder GFP in the presence of clindamycin. As you can see, wild type does not do very well. It doesn't produce much GFP. Our control genotype, a known clindamycin resistant genotype, does produce GFP in the presence of clindamycin. And then if you look at our library of variants selected in the presence of clindamycin, they actually do even a little bit better than that control variant. We took the top 20 of the genotypes that were selected in the pool, individually cloned them, and expressed GFP in the presence or absence of clindamycin. And you can see that most of them are functional. 11 out of the 20 are more functional in the presence of clindamycin, or they produce more GFP in the presence of clindamycin than our control mutants. And a handful of them have really comparable activities to wild type in producing GFP in the absence of clindamycin. So this is encouraging. We really want to be able to see that not only does the selection look consistent, but the things you pull out the other end actually do what you expected them to do. I also wanted to look at the selection dynamics within a single round. So I took the library and I did a round of selection in the presence of clindamycin. I took out samples and I quenched the reactions with tetracycline at time points between 7.5 and 90 minutes. And I basically looked at how well the enrichment of genotypes at each time point correlated with the known clindamycin resistant genotypes that we got out the end of the selection. So if we got a weak correlation, as you see on the left there, that means that at that time point, we aren't getting great enrichment for clindamycin-resistant mutants, whereas a strong correlation, as you would see on the right, would mean that we are getting good enrichment of clindamycin-resistant mutants. So when I look at all the time points together, it looked kind of interesting. Really surprisingly, it looks like we're building functional ribosomes and actually translating our selective peptide within 7.5 minutes. We're already getting successful ternary complexes and a very good stringent selection. Interestingly, that kind of falls off, and then in the middle time points, the selection is not very good. We've speculated about why that is, but we're not sure. And then you get a stringent selection ramping up again after 70 minutes. So we kind of decided that really the longer incubation is the way to go, because you don't want to miss this optimal time at the very beginning here and end up in this kind of weak selection regime. So we think that incubating for a longer time is the best way to go. Another interesting thing is that winning genotypes do not always they're not always the ones you would necessarily expect. So the consensus genotype here, based on what was the most frequent nucleotide at each position, is ATG-AGA. This was a very fit genotype, but it was actually present at about 1/3 the frequency that you would expect, given just the frequency of each nucleotide. Meanwhile, these other two genotypes were present at about 8-fold and 18-fold the frequency that you would expect, just based on the positions at each nucleotide. So this sort of hints at something that a lot of evolutionary biologists think is interesting and an important force in evolution, which is the question of epistasis. So epistasis this is the equation that we use to calculate epistasis in our experiment. And basically epistasis is equal to the log of the observed fitness of a given mutant. So we observed this in our selection, divided by the expected fitness given an assumption of independent contribution of each position to fitness. So with this equation, positive epistasis is values greater than 0. And these are genotypes which are more fit than you would expect, given an independent assumption of contribution to fitness. And negative epistasis is values less than 0, or genotypes that are less fit than you'd expect. So by calculating epistasis values, we already have fitness values for our mutants. And we also have relatedness. So we can look at all the genotypes and look at how they're related to each other and which ones are individual point mutations away from the other. So with this information, I was able to build a network plot. You can kind of think of this network plot as just an image of the fitness landscape, all of the information in one place. It can be a little difficult to interpret, but you can kind of just look at it and see some general trends. You can see, for example, neighborhoods of positive epistasis. These genotypes, which are kind of closely related to each other, many of them display sort of mild positive epistasis, or in the upper right-hand corner too, you see a little neighborhood of positive epistasis. Another important thing to note about this plot is that it's a very densely connected network. You know, you have a few genotypes on the outskirts here, which are not related to very much, but most of them are closely related to each other. So this sort of hints at the idea that we're selecting related genotypes because those genotypes have related function. All of this just driving at the point that our selection is really coherent and really selecting for a common phenotype, which is translation ability in the presence of clindamycin. Just wanting to sort of more quantitatively look at this stuff a little bit. I built a histogram of the epistasis values of starting genotypes in gray versus the winning genotypes in blue. So you can see that the winning genotypes are substantially enriched for positive epistasis. So this to me suggests that positive epistasis is really important in determining the most fit genotypes in a population. And that's important for directed evolution because positive epistasis makes it kind of difficult to necessarily predict which genotypes are going to be the winners. So this is why directed evolution is important. Rather than try to predict from a set of point mutations which combination of those point mutations will be the most fit, we just create a large library and select the best genotype out from it. Another thing that we noticed, so edges per node is a measure of relatedness of a given genotype to other genotypes in the winning population. So you can see here, this node, which is this point here, has 6 edges. That means that it is a single point mutation away from 6 other genotypes that are in the winning population. If you graph edges per node against enrichment, or basically fitness, you get a pretty good correlation in that genotypes which are more related to others in the population are in general more fit. So again, this just hints at that our selection is sort of coherent and selecting for a common phenotype. So then, of course, an important question in all this epistasis business is whether or not these genotypes which display extreme positive epistasis are actually functional. You know, we've seen that our very best genotypes are functional and do make protein in the presence of clindamycin, but are these very strong positive epistasis variants, are they functional? So we cloned some of these genotypes with the highest positive epistasis value, and the answer is yes and no. So some of the ones with the very highest values, sort of to the left of the box here, these are mostly not functional, but a few of them are. And these are things that if you looked at, you know, sequence alignments from ribosomes that exist in the wild, you would never, ever build this sequence because you would never predict that it would function. But these ones actually do. And this highlights that really the ribosome is actually more amenable to mutation if all you're interested in is translational activity than we maybe thought before, because many mutations which will kill a cell don't actually destroy translation. They're just not compatible with life. So I talked about trying to, you know, use the ribosome to make non-canonical polymers. Obviously, I haven't shown any data about that today, but sorry. We are thinking about that in the future. That's the next direction that we would like to go with this. Oh, just takeaways from the RISE platform. We can rapidly select ribosomal RNA variants. The interselection dynamics are super important for specificity and strength, and you really need to sort of determine those for any given selection you're doing. And then the consensus selection sequence is really of limited utility in predicting the winners. So you need to actually do the evolution and find out. In the future, we're looking at making interesting ribosome libraries of the exit tunnel, the peptidyl transferase center, all these parts of the ribosome, to try to figure out what are the important areas to mutate and to select on to enable new function. So here is just a model of the exit tunnel with all of the nucleotides that surround it. We think the exit tunnel is going to be an important target because it's very, it's well evolved to accommodate L-alpha amino acids, but it's not evolved to accommodate polymers of other types. So we're currently exploring how we can use this platform to study epistasis in more depth and to engineer the ribosome for new function. So that's it for me. Sorry, I went a little bit over. I just want to thank everybody who's helped me with this on Team Ribosome, and for Mike Jewett. I'd like to thank Mike Jewett for creating the environment where we can make all this cool stuff happen.
Thank you. Thank you very much, Mike.